Good afternoon, everyone, and welcome to the Front Row Lecture Series. It's my real pleasure to introduce today our President and CEO, Pete Schultz. I think we're all looking forward to a hopeful 2021. Vaccines are starting to roll out. Some of the people on campus have been vaccinated, and I think there's grounds for optimism for us to return to normal in the coming months. So we're really looking forward to that. We thought it would be a great opportunity to have Pete launch off on the 2021 Front Row Lecture Series and give us an update on what's happening. So let me first say that it's always a little challenging to introduce your boss. I think he's probably nervous that I could provide some embarrassing stories, and I, of course, am nervous that I might do something untoward. So I think we're just going to leave it at mutually assured destruction as possible, and we'll just proceed on to the main event. So let me just give the briefest bio for Pete. Pete got his PhD at Caltech with Peter Dervin and did a postdoc at Harvard with Chris Walsh before joining the faculty at UC Berkeley. That was, I think, around 1985. And Pete rapidly rose through the ranks. And I recall watching this. I was a postdoc hopeful wanting to join academia, and Pete was just a phenom and rapidly became a full professor. Now, Pete moved to Scripps roughly 20 years ago. And at the same time he came to Scripps, he founded the GNF, the Genomics Foundation for the Novartis Institute, which is located right next to our campus. And after maybe 10 years working on that, he started an independent biomedical research institute called the Caliber. And when Pete became the president of Scripps in 2015, we worked on merging in Caliber. And so now Caliber is actually part of Scripps, and it is drug discovery in a not-for-profit setting. And Pete is going to tell us today about the latest updates in trying to rapidly generate new medicines in an academic setting. So without further ado, I'm going to introduce Pete. So thanks, everyone, and enjoy the lecture. Pete.
Great. Thanks a lot, Jamie. To all of those who heard my previous front row lecture, I'm here to give you a progress report on how we're doing. And for those of you who haven't heard an update before, I thought I'd give you an overview of the model we're building and where we stand in building that model. When I took over the leadership of Scripps, I asked whether we could do something kind of new and potentially transformational with biomedical research institutes. And that's not only to advance scientific discovery and technology development, which Scripps has a rich history of, but to accelerate the translation of those scientific discoveries to new, innovative new medicines that directly impact patients. And by bridging basic research and translational research, we also want to create a self-sustaining new quote-unquote business model for nonprofit research that provides revenues that can be reinvested in the research enterprise. So that was the vision for Scripps. Just a little bit of background on Scripps for those who may not know Scripps. It's one of the largest nonprofit research institutes in the U.S. It has two sites, one in La Jolla and one in Jupiter, Florida. I tend to have an aversion to self-promotion, but I have to say that Nature, one of the most prestigious scientific journals, ranked Scripps as the leading nonprofit research institute in the U.S. And in fact, number one in terms of its impact on scientific innovation across all universities and institutes worldwide. So we are the best kept secret, but we're trying to change that. We We've also had a large impact on the public. We have 10 FDA-approved drugs that in some ways Scripps contributed to, and 80 plus spin-out companies. And something we're really proud of is having one of the top 10 graduate schools in the US. Scripps, as I said, has a rich history of scientific discovery and technology development, from understanding how you sense external stimuli, temperature, pain, touch, to understanding the molecular basis of how the immune system recognizes self versus non-self, to new technologies in proteomics, drug discovery, vaccine design, and to new methods for making molecules, chemical methods and biological methods. But despite this rich history here at Scripps, and in fact throughout the academic world, it's the golden age of science in terms of discovery, we still have an overwhelming need for new medicines. From degenerative diseases of aging to cancer to infectious disease. And the question is why? Why, if we know so much about human biology, are we still slow in translating that to new medicines? And the answer may be that most discoveries are made in academic institutions, which really don't have the infrastructure nor the experience to turn those discoveries into new drugs. New drugs are made in biotech and pharma. Biotech and pharma and academic institutions have very, very different cultures, motivations, processes. And those differences have historically created a lot of intrinsic barriers that slow drug discovery down and make it a more costly process. So what we asked is can we actually accelerate that by bringing basic research, drug discovery, and clinical medicine in patients all under one roof. So we really do go all the way from the laboratory bench to the patient's bedside. And how do you do that? And we thought by doing that and making that process seamless, we could really accelerate the development of new medicines. So one could build drug discovery and early stage clinical capabilities from scratch, but that would be time consuming and quite costly. What we did was to go a different route. We actually created a drug discovery division by making Caliber, a nonprofit research institute, part of Scripps in 2018. Caliber was founded in 2012 as a nonprofit research center focused on drug discovery and early stage clinical trials. That was its mission. I happened to be president of it, so when I took on the leadership of Scripps, we combined the two and instantly came up like a square wave with a drug discovery and clinical research capability. Caliber is funded by major foundations, a large collaboration with the Bill and Melinda Gates Foundation, Wellcome Trust, also by federal and state agencies, CIRM, NIH, and by pharma, most recently with AbbVie. It has extensive preclinical and clinical drug discovery expertise and infrastructure. We work in many areas of medicine, from cancer all the way to metabolic and infectious disease. And importantly, we're a highly collaborative, have a highly collaborative approach towards translational research. We do have, as this slide shows, state-of-the-art capabilities that bridge almost all aspects of early stage drug discovery and development, from protein engineering, cell therapies, small molecule medicinal chemistry, pharmacology, IND-enabling studies, and early clinical capabilities. And as I said, we have collaborations throughout the world with major medical centers, San Diego, LA, Cincinnati, with corporate partners, with major foundations, and with research institutes and universities throughout the world. We really do believe that you can't do everything yourself, so why not partner with the best and brightest and work together to advance science and medicine. So what's our focus? We tend to be a little arrogant. Rather than work on just one disease, we actually think we can tackle many diseases that represent the world's most pressing healthcare challenges. These include diseases of aging that really negatively impact the quality of our lives as we get older and the costs of healthcare. Chronic and childhood diseases that impact quality of life throughout life and also are a major healthcare cost, all the way to infectious diseases, which can have a global impact, as we saw in the last year with COVID. Our approach is to pursue innovative approaches and technologies that really complement what's being done in industry. These, for example, regenerative and reparative medicines, harnessing human biology to fight disease, tissue-targeted therapies, lower cost repurposed medicines, and the like. And in particular, our focus is really determined by unmet medical need rather than financial considerations. So where are we? What kind of progress have we made in the last few years to realize this vision that we can bridge basic research with the development of new medicines? Well, this slide shows our pipeline just at a high level. We have 4 programs that we've moved into the clinic ourselves, and 2 with the Gates Foundation, which is really unprecedented for a nonprofit research institute. And we also, with our pipeline, are positioned to move 2 molecules, 2 or more molecules into the clinic every year for the next few years. So we've made a lot of progress, and what I thought I'd do is describe a number of these programs with small vignettes, just to give you an idea of the nuts and bolts of what's going on. As I said, one of our major focuses is regenerative medicine, reparative medicine. And this is really a kind of paradigm shifting approach. It's not to make drugs that slow the progression of disease, but it's to make drugs that actually reverse the disease process. And how do you do this? Well, the idea then is to make drugs that work on endogenous stem cell populations that are already in all of us. We have neuronal stem cells in our brains, hematopoietic stem cells in our blood, and so forth. So the idea is to make molecules that impact those stem cells and control their fate, to repair damage. And our focus is repairing damage to joints and major organs. So we have 2 programs developing drugs for osteoarthritis, another for meniscal repair, another protein biologic for treating arthrofibrosis, which is frozen joints, a program for wound healing, for diabetic wounds and burns, liver regeneration and cancer treatment, lung regeneration for chronic lung disease, and remyelination in MS. And just a couple of examples. One of the programs that's in clinical trials right now is a drug, the goal of which is to regenerate cartilage in OA. 40 million suffer from severe OA in the developed world alone. I'm sure many on this Zoom call probably have knee replacements. And the idea of this approach is, can you actually repair the damage rather than needing a knee replacement? Can you actually make new cartilage? So to that end, we took stem cells that are resident in our knees and carried out, using our large chemical libraries and screening technologies, a screen to look for molecules that would turn those resident stem cells in our knee, those are called MSCs, mesenchymal stem cells, into chondrocytes. And chondrocytes are cells that make cartilage. So if you have a drug that makes chondrocytes, you have a drug that makes cartilage. And we found such a molecule. It operates by a unique mechanism. It works in preclinical animal models, and we actually have put it into patients as an intra-articular injection. So we completed a phase 1 safety study, and the drug is safe. And now we're looking at creating an extended release formula to test in a larger phase 2 clinical trial for efficacy. One other example is repairing the lung. There are many chronic lung diseases where you slowly lose lung function irreversibly. For instance, COPD, idiopathic pulmonary fibrosis. The life expectancy is 2 to 3 years. And there are other diseases such as respiratory viruses, silicosis, and others. So there are no therapies that correct the damage. And how would you do that? Well, again, we have resident stem cells in our lungs called AEC2 cells. So we put these stem cells in a dish and again screened our library for molecules that would expand or proliferate those stem cells to make more of them so they could then subsequently turn into lung. We found a molecule. It works well in animal models of lung disease. It actually makes new lung. We've shown that. And we've actually now made a lung-targeted formulation of that molecule. So the idea is you inhale the molecule and you actually begin to regenerate lung. And our first, we're moving that program forward into to the clinic, first for IPF, pulmonary fibrosis, but we think it could be applicable to a wide range of pulmonary diseases. We also have a major focus on cancer. We have a focus on immunotherapies for cancer, which include bispecific molecules, CAR T cells, STING and gamma delta T cell agonists. And then we have targeted therapies that target kinases involved in cancer, and even repurposing efforts for drugs for myelodysplastic syndrome. And just a couple of examples here. One is a cell therapy for cancer. It's called CAR T cells. This is a therapy where one takes out of a patient T cells, killer T cells, which are the cells that attack virally infected cells and other cells in the body. And you genetically reprogram those cells with a receptor that targets them specifically to cancer. So when you put those killer T cells back in the patients, they attack the cancer cells. And the results from others have been remarkable. 90% remissions in patients with relapsed refractory leukemias. The problem is it's a living drug, so you can't control turning the T cells on or off. So we created a 2-stage CAR T cell therapy, which incorporates into the T cells a switch, which means you can step up the activity of the T cells or turn it off. It's like turning a switch. And so that therapy is now in patients as we speak. We're testing that in patients with lymphomas and leukemias. And we're optimistic that if this works, it will overcome the safety hurdles that have prevented the expansion of CAR T cells to solid tumors like breast cancer and prostate cancer. Not only that, we think being able to control the CAR T cells will improve their therapeutic efficacy and also make them more available to the general community. We also, I should say, have a great partnership with AbbVie working on applying this technology to breast and prostate cancer. We also are taking a different approach towards using the body's immune system to kill cancer. We've made 2-pronged molecules that we've designed, where one end of the molecule grabs onto a killer T cell and the other end selectively grabs onto a cancer cell, brings the two together, and when the T cell is in close proximity to the cancer cell, it kills it. And what's clever about this is we actually use the imaging agent that's currently used to image prostate cancer is a targeting agent to target the T cells to prostate cancer. And our focus right now is a clinical trial on metastatic prostate cancer. It's the second leading cancer in men, prostate cancer, and in 10 to 20% of patients with prostate cancer develop castration-resistant prostate cancer that becomes metastatic. And right now there's no treatment for that. So we're optimistic this drug that again uses T cells will actually provide a new treatment for people with metastatic prostate cancer. Another approach we're taking to harness the immune system to kill cancer is molecules that generally turn on the immune system systemically to kill cancers. And this takes, one of these approaches takes advantage of the cGAS-STING pathway. This is a pathway that when a virus infects you, it turns on this pathway, activates your immune system to fight the virus. Well, there's a realization that if you make drugs that turn on this pathway, you can actually turn on the pathway to fight cancer as well. And drugs have been made that activate this pathway, but they have to be directly injected into the tumor, which really limits them to a very narrow spectrum of cancers. So we've again used our drug discovery infrastructure, our high throughput screening robotics, to find molecules, small molecules that activate these pathways, and we've used structure-based design to actually optimize those molecules. And so we hope actually to have a clinical candidate sometime this year, which again could be very useful for a broad range of cancers. Another focus of our efforts is in neurodegenerative diseases. That I'm sure everybody on the Zoom knows somebody who has either Alzheimer's, Parkinson's, ALS, or some debilitating neurological disease. We're taking multiple approaches to neurodegenerative disease. And again, the common theme here is exploiting the body's own biological mechanisms to fight disease. In one case, we're taking advantage now of the immune system to prevent disease. In other cases, we're taking advantage of other pathways, autophagy pathways, to eliminate toxic agents from the brain, ARE activators to turn on antioxidant or protective mechanisms. And we're even making long-acting drugs for CNS disease, like long-acting seizure medicines. So just by way of a couple of examples here, one approach for treating neurodegenerative diseases is a neuroprotective strategy. It's clear that neuroinflammation plays a major role in degenerative diseases like Alzheimer's and Parkinson's. And one way to combat that neuroinflammation and protect neurons is to suppress the immune system. And here we use another kind of T cell called a regulatory T cell that actually attenuates the immune response rather than activating it. And so it's been shown in animal models and also in two small clinical trials that if you expand Treg cells, you actually slow the progression, stop the progression of ALS, and actually the same thing's true of Parkinson's disease. So how do you do that? How do you expand Tregs? Well, there's a natural protein, GM-CSF, that does that. The problem is it has a short half-life. So if you want to use it to make more Tregs, you have to take injections every day. So we used a platform technology we've developed to create an antibody GM-CSF fusion. And this molecule has a long half-life. It's incredibly potent, GM-CSF. But we think it can be dosed once a month into a patient. So it would have a huge impact, we think. So we've moved this molecule forward, and right now we're looking at starting IND-enabling studies in manufacturing for clinical trials, both in Parkinson's disease, and we're also talking with the Seeley Center at Mass General to carry out a trial in ALS. Another earlier stage program is harnessing the cell's recycling systems to fight neurodegenerative disease. It turns out protein aggregates, precipitated proteins in the brain and damaged organelles are highly toxic to neurons. So people have been trying to clear this damage for years as a way to treat neurodegenerative disease. Well, there's a natural mechanism for recycling garbage in the cell. It's called autophagy. So if you can activate that natural mechanism, it might be very useful in removing neurotoxic agents from the cell. So again, we've used our technologies to set up screens to look for molecules that activate autophagy, in this case, simply by removing oil droplets from the cell. And we found molecules, and they actually clear protein aggregates in the axons of primary neurons. So we're really excited about also pushing this approach forward. As I said, another focus is chronic diseases. These are diseases that affect the quality of people's lives throughout the entirety or for a large portion of their lives, and actually impact the healthcare system very much. So we're focused on disease reversing therapies here for intestinal diseases like Crohn's disease and ulcerative colitis, for obesity and diabetes, for respiratory diseases, and for fibrosis. And just again, two examples here is the first I'll focus on is intestinal bowel disease. Just in US and Europe alone, 3 million people suffer from IBD. The way we treat IBD today is with anti-inflammatory agents. But a part of the disease itself is the intestinal barrier that separates all of the microbes in your gut from the rest of your body is compromised. So you get leakage, and that leakage leads to inflammation. So if you really want to impact the disease, you want to repair that barrier so there's no leakage. There's a natural peptide called GLP-2 that's an intestinotropic peptide that actually stimulates repair of the barrier. People have used that to treat certain diseases like short bowel disease, but its half-life is too short to really be used in IBD. So we've used, again, technologies we have to engineer natural peptides to make them highly potent and long-lived. And we've made such a version of GLP-2. And you can see on that slide on the right, the natural barrier is in the top left-hand picture. In a disease model, you lose barrier function in the top right, and when you give the drug, you restore it in the bottom left. So we're moving this molecule now into IND-enabling studies for treatment of Crohn's disease, and that's in partnership, likely partnership with AbbVie. Another disease that impacts a huge number of people is fibrosis. It's fibrosis of the lung, fibrosis of the heart, and fibrosis of the liver, so scarring, and you lose function when you scar your organs. And really, there are no really effective anti-fibrotic drugs. So we've been searching for new drugs and new strategies to treat fibrosis. And again, the approach we're taking here is to make drugs that work on cell types that lead to fibrosis. So one event that leads to fibrosis is a differentiation of a cell called a myofibroblast into a fibroblast. So we set this up again in a little 1536 wells, and we looked for molecules that blocked that cell transformation. And we found one, and surprisingly, it was an antifungal drug. And we were confused, what's an antifungal drug having antifibrotic activity? And in fact, there was an anecdotal report in the literature, medical literature, that suggested this antifungal would resolve keloids and scars, fibrosis. So we actually learned that these antifungals have a completely separate activity and mechanism distinct from their antifungal activity that actually blocks fibrosis in animal models. So now we're carrying out a large phase 2 study with Cincinnati Children's to reproduce the anecdotal evidence in the literature in kids with keloids and scars, which can be really traumatic if they're disfiguring. And if that works, we've actually made in parallel a molecule that has an improved pharmacological profile for treating chronic diseases like IPF or like fibrosis of the heart. And finally, we have a large activity in neglected infectious diseases. And this is in collaboration with the Bill and Melinda Gates Foundation, and more recently, the Wellcome Trust. We've become their hubs for drug discovery efforts. And those drug discovery efforts are through a worldwide network of collaborators who are focused on infectious diseases, including HIV, tuberculosis, malaria, COVID-19, and hepatitis. As part of our global health initiative, we also have a large effort using genomics tools to transform our ability to detect and respond and forecast pathogenic outbreaks like COVID. And we also have a rational vaccine design group, one of the largest in the nonprofit sector, that's working on using structure-based tools to design new and more effective vaccines for HIV, influenza, and COVID. So just as part of our Gates Global Health pipeline, we've actually put 2 molecules in the clinic, one for childhood diarrhea and one for TB, and we have 2 more going into the clinic, we think this year, one for HIV and one for malaria, and then a pipeline of candidates behind that. One example is a drug for HIV. We actually worked with the Gates Foundation to find a drug that's a small molecule drug that's been engineered so you can give a subcutaneous injection, we hope, once every 6 months to either treat or, just as important, prevent the spread of HIV. So instead of a yearly flu shot, it's a bi-yearly shot that protects you from HIV. So this molecule looks really good. We've taken it all the way through non-human primate studies, and it's on track for the phase 1 trial in healthy volunteers in 2021, early 2022. So we think this can have a dramatic impact on HIV. We're also working on TB. TB kills 1.5 million people a year, every year, and there are no good vaccines for TB. The problem with drug therapies is they take a long time to sterilize the patient, and they also create the potential for drug resistance. So in collaboration with a group at Cornell, we've been screening for molecules. It's clear that a combination of drugs is really required to effectively treat TB. We've been looking for drugs which, when combined with the current drugs, potentiate their action. And we were actually able to find a molecule that activates an enzyme called adenylyl cyclase, which is in cholesterol catabolism, that when used in combination with 2 other TB drugs, sterilizes the mice of TB, which is really, really quite exciting. So we're right now working on moving that with the Gates Foundation into testing in human patients. And then another approach, we've been working with the Gates Foundation on endectocides. These are drugs that when you take the drug and the vector that carries the disease, like the mosquito that carries malaria, bites you, it ingests the drug and dies. The drug is safe for you, but it kills the insect vector. And there's been modeling that suggests an effective drug could block transmission 90%. So we're working on that kind of a drug. But we found a really good class of drugs that does that with malaria vectors, mosquitoes. But we realized it also works with ticks. And the beauty of this drug with ticks is that it actually kills the tick before it transmits the disease. So the idea is, if you want to go hiking in the woods in New Hampshire, you take a pill before you go hiking, and any tick that bites you dies before it transmits Lyme disease. And it turns out this class of drugs is already used in dogs, and the FDA just approved it as a preventative treatment for Lyme disease. So our dogs run around in the fields and don't get Lyme, but we do. So we're working very hard on making a drug for human use with ideal human pharmacology. And we hope to have a candidate this year, as a preventative treatment. As I said, we have one of the largest vaccine development groups in the nonprofit world. Right now, we have two HIV vaccines in clinical trials and a third going through manufacturing. We're also working on vaccines for influenza and COVID. We also have the largest drug repurposing collection ever built in the world. This is a collection of every drug that's ever been put into people in some late stage preclinical drugs. And the beauty of repurposing is if you find a drug that's, say, an antifungal that works for fibrosis, you can rapidly move that drug into people because we know its safety and its pharmacology. And, there are many examples. Thalidomide became a drug for multiple myeloma. It was repurposed. So we've built this collection. We collaborate with labs throughout the world to find repurposed drugs for neglected diseases. We'd like to expand that to all diseases. We've identified more than a dozen such drugs. And we've become the hub for the distribution of this. So when COVID came along, we actually mobilized Reframe and sent it to over 30 labs around the world, collaborations with pharma and with universities and institutes, and screened this collection for drugs that could be repurposed for COVID. And a number of those drugs are currently being tested in animal models for their effectiveness as single agents and combinations. But we've also found some drugs that work okay. And that's a good way to make a drug that works well. So we've also initiated a large effort to optimize the repurposed drugs we found for COVID. And in fact, we've built a really quite comprehensive drug repurposing effort. A quite comprehensive COVID-19 research effort at Scripps. It involves, as I said, using genomics and wearable devices to monitor the spread of COVID and understand the mechanisms by which it spreads. Efforts to create vaccines to prevent the disease. In fact, our labs were the first to solve the crystal structure of the coronavirus spike protein, and that allowed engineering stabilizing mutations to be made that are used right now in the Moderna and Pfizer vaccines that some of you have gotten. And as I said, we also have a large effort to identify antivirals and create antivirals. But there's an important, we're dealing with COVID right now, but one of the major lessons we've learned is maybe we should be preparing for the next pandemic. It will happen. There will be another pandemic virus. It could be another coronavirus. It could be influenza. It could be other viral families. And why don't we, we've spent a huge amount of resources preparing for military conflicts, potential military conflicts. We just fought a war against, we're fighting a war against the small little virus, and we're not doing so well. So the idea is, should we begin to invest resources in preparing for the next pandemic, making antivirals that work against the next likely pandemic viruses, which we can probably begin to identify. So that's also a major effort and direction we're going in, again, with collaboration with foundations like Gates and others. And finally, I should point out, and you've heard a front row lecture about this, we also have the Scripps Research Translational Institute, which was founded by Eric Topol and is led by Eric Topol. And the focus of this institute is really to understand disease at the level of individuals. You know, how does cancer affect me differently than how does it affect you? And this is being done by using genomic tools, by using wireless medical technologies to create databases and the like. Eric's group is running the largest NIH-sponsored personalized precision medicine initiative, a $250 million initiative. They've also, another example is they've created PowerMom, which is using a digital approach to create a database of pregnancy-related health issues to really inform us on those factors that affect maternal health outcomes. So where are we? I think we've validated the idea that you can seamlessly bridge basic research with drug discovery and even with early stage clinical development, where we're actually taking drugs and treating patients, directly impacting patients. I think because this approach has been validated with some of the examples I've given you, the idea is now we can scale this, knowing that it works. I think it's scalable. And we can scale it in a number of ways. We can scale it by expanding those diseases we try and discover drugs for. Our neglected and infectious disease efforts are funded by major foundations, Gates and Wellcome Trust. The other activities in chronic diseases, cancer, and diseases of aging are funded opportunistically. So creating the same kinds of resources there is a priority for us. And actually expanding our ability to put these drugs into patients is a major direction we want to go, which means we need to build more expertise and capabilities in that area. But that's where you do want to go. You actually want to make a drug and show that it actually has an impact on patients. So that's the overview. What are we trying to do? As I said, we're trying to pioneer a new vision for nonprofit research and show that nonprofit research institutes in these changing times with these changing needs can not only do great science, but can accelerate the impact of science, great science and technology on public health. And I think we've started to do that, and we're really quite optimistic we can expand those efforts. I should point out Scripps is unique. It's a highly entrepreneurial institution. We actually invest a lot of our resources into high-risk reward opportunities to advance science and medicine, some of which I described here today. And that's a bit different than many other institutions that tend to be more conservative and invest in building an endowment. It's an interesting approach for universities. If you invest in a company, an outstanding company doesn't put the investment into building an endowment. It puts the investment into scaling its impact and creating a multiplier of that investment. And the interesting question is, why don't nonprofits do the same kind of thing? It means you have to take some risks, but you can really amplify the impact of resources going into science. And so as part of our success in our investment in science and creating new drugs, we hope as we partner those drugs, successful drugs, with pharmaceutical companies for further development and approval, that the revenues that are returned to us can be reinvested in science and in drug discovery to really create a self-sustaining model that not only sustains itself, but grows with time to have an even bigger impact. So that's the story, and that's where we're at. The last thing I need to do is thank all of you for your enthusiasm and for your support that makes our work possible. We just have, we're really delighted with the increased involvement of the community with Scripps, and it's really a delight. And I'd be happy to answer any questions, Janie.
Okay, thanks, Pete. Thanks for a terrific lecture. We've got quite a few questions queued up. Let's just start with one. You were talking about a regenerative lung tissue treatment. And so the questions come up, will that be applicable to people who have, lung damage from COVID-19?
Yeah, that's a good question. We actually think it could be, okay? We think the first best test case is pulmonary fibrosis, given the nature of the disease and the nature of the cell populations that are lost. But we think this could be useful for COVID. We think it could be useful for silicosis. And also it potentially could be useful for COPD. On another front, we've actually made another lung-targeted drug that we really are quite interested for COVID. When you get COVID, neutrophils invade your lungs and cause a huge amount of damage. And we've seen that dexamethasone, actually, it's a known anti-inflammatory, actually is useful for treating COVID-19. We now have a small molecule that targets damaging proteins that are made by neutrophils that might complement dexamethasone and actually have a significant impact on lung recovery when used with COVID patients. So both abrogating the impact of the disease on lung and potentially with the lung regenerative medicine repairing the lung.
Right. So here's more of a philosophical question, and I'll paraphrase it. But so what are some of the problems and obstacles that you've had to overcome by taking an academic research approach and then using that to translate medicines into real discoveries, and what's the hard part? This is an unusual program. What was the biggest challenge?
Yeah, so the reason we thought we could do this, the biggest challenge was nobody thought we could do it, except for a few people. And the reason we thought we could do it is when I moved to Scripps, I spent some time actually creating a genomics institute with Novartis, which wound up turning into a drug discovery institute. And Novartis, over the 10-year period we built and ran it, gave us about well over $1 billion to build tools for drug discovery and apply those tools and learn how to use them. And it was really successful, but Novartis had enough money that we could pave over our mistakes simply by asking Novartis for more money. Okay, in the nonprofit world, you don't have those kinds of resources available to you. So that prior experience was incredibly helpful. I think without that, we would have made mistakes doing it in a nonprofit, and we couldn't have survived those mistakes. So that really, I think, was a huge enabler for us to do this, that other people didn't have the experience of that learning experience. And then the rest of it was just people saying, there's no way you can do this, okay? And by the way, you shouldn't work on more than one disease, okay? But luckily, I had worked in a previous life with the president of Gates Global Health. And he called me on the phone and said, wow, now that you're doing this, let's form a partnership. And the Gates Foundation and Bill Gates have been incredibly supportive. And a good friend of mine at the time ran Merck and had seen what we'd done, and Peter Kim, and he also decided to place a bet on us. So we had a couple of daring people help fund the beginning of this, and that's how it happened.
So Pete, you touched on this a little bit about preparing for the next pandemic. And I thought it might be worth kind of going through the challenges, like who are all the players involved? And I know Gates was, predicting a pandemic, and so it's really on their radar. And then we have various governmental agencies, and then the World Health Organization. So what's the challenges and what's the interplay?
Yeah, so the challenge is who's on first, okay? So just actually, there are a lot of folks interested in okay, we didn't do very well this time. What can we do to position ourselves better next time? But nobody is really leading the charge on this, and really just organizing the effort. And it needs to be an integrated effort. We need to be able to detect these viruses quickly and the spread of these viruses. We need to be able to develop vaccines, and no matter what, that's going to take a year or two, if possible. And we need antiviral drugs, and we need all three of those. And we can actually probably predict, the Gates Foundation has laid out the next likely three viral families that will cause pandemics. And interestingly, many of the viruses in those families are quite related in the proteins that you want to target to kill the virus. So the idea is we can start to prepare kind of archives of advanced medicines that target, say, Mpro and coronaviruses, okay, or TMPRSS2, or the polymerases, and have those ready in all of the BSL-3 laboratory facilities, the animal models, the molecular biology, all ready to go. So we can actually screen and probably put drugs into people within the first few months, which even, with COVID-19 and the vaccine, there's been a huge number of people that have died while those vaccines were developed, and they were rapidly developed. So it becomes an organizational issue, and just, an issue of the world deciding that this is a really important priority and investing. It's not going to take trillions of dollars. It's on the order of single-digit billions of dollars to make sure this doesn't happen again, or to really lower the probability that it's going to happen again.
Great, yeah, thanks. I have another question that I'll paraphrase. I think what may not be apparent to the audience is that all of the remarkable work you talked about in the past hour is really carried out by about 100 scientists at Caliber. And so there's 1,000 other scientists at work in various departments at Scripps, and so maybe you could just touch on what they're up to, and then the relationships between the basic research and the Caliber.
Yeah. So when we started to have this vision for Scripps, unfortunately, I'm 64, so you don't want to fool around to actually have an impact. So we really wanted this to come up like a square wave. So that's why we brought Caliber into it. And that's why many of the programs you saw were initiated at Caliber. But what's happened now that Caliber's become part of Scripps, we have many, many collaborations. For instance, the lung regeneration program was, one of the young assistant professors here has played the major role in moving that program forward. The autophagy program, a senior chemist is actually pushing that program. The vaccines and adjuvants are all being done in collaboration. The STING agonist came out of an assistant professor's lab. So what we're seeing is a lot of faculty want to see the impact of their basic research on human health. And now that, you know, people are realizing these capabilities are available, they're starting to use them. But, you know, for every drug that's developed, you need to develop the underlying biology, the underlying technologies. So there are labs at Scripps that have played a major role in proteomics and genomics technologies, okay, in developing, you know, DNA encoded libraries and other tools that are all used to enable and facilitate translational research, and also just understand the underlying basis. The crystal, as I said, the structure of the spike protein was solved using modern structural tools very, very quickly by people who are also solving a lot of other fundamental problems. So it's beginning to come together as a large, very cohesive, as I said, seamless community. And I think, you know, that's really exciting. And what I can say is scientists of the current generation, and some in my generation even, are becoming more focused on not only doing great science, but actually helping to impact public health with that science. So they love to see, you know, their science turned into a drug that helps the patient.
Another question that's come up is, you know, concerning artificial intelligence and AI. So what do you see are the opportunities for us to, you know, bring these powerful new technologies to bear on science and drug discovery?
Yeah, I think there's a huge opportunity there. You know, Eric Topol has been involved in understanding and a strong proponent of the impact of AI in medicine. And I think Scripps is unique in the sense that we have many potential applications for AI. You know, there's very clear applications in determining protein structures and understanding, you know, gene transcription, what genes are turned on, and so forth. There's probably a major opportunity in understanding the data that's coming out of large-scale libraries and the databases like PowerMom and others. So that's a very, very exciting future direction, and understanding, you know, the challenge I think there a bit is understanding those problems that are ready for AI now, where you can have an impact and show the impact, versus those problems that will be ready for AI 5 years from now. So really understanding where to start and where you want to wind up 5 years or 10 years from now is a key issue there.
So I know you touched on this briefly, but maybe there's a couple people that are curious as how do you actually, you know, what's the next steps for these programs to bring them to market? Clearly you can't do it all in Caliber. So how are you going to spin out companies? Are you going to license? What are the options that you're looking at to
So that's a really good question. We kind of have created a pipeline and moved molecules into the clinic, but we have many, many more opportunities to move molecules into the clinic to directly impact patients. And that becomes expensive, and you need a lot of expertise to do that. So we're right now working with, we have two boards at Scripps, a board of overseers, which involve a lot of people from biotech and pharma that are really interested in the model we're building, as well as, you know, the investment communities, as well as our board of directors, which also has people very much involved in life sciences and business. We're working hard to really develop a strategy for how best to expand our ability to move the pipeline we've created already further into the clinic. We don't want to do what pharma does well, which is later stage clinical trials, which are large and very expensive. Rather, we'd actually like to have as our endpoint moving drugs into patients and showing they impact disease. And once you show they're not only safe, but impact disease processes, either in phase 1 or phase 2A trials, then forming a partnership with pharma who has the resources and expertise to do larger phase 2 and phase 3 trials makes sense. So that's the model, is that's where we want to stop, but it is expensive. Some of these phase 1 trials cost $20 or $30 million. And how do you do 10 of those? And so we're looking hard at all kinds of different pathways to do that. Funding from foundations. CIRM moved our OA molecule into phase 1 trials. Gates has done the same. And from pharma partners who are really interested in our programs, to NUCOs, or to even creating a fund for Scripps that invests in these opportunities, where not only do you impact disease by facilitating, clinical studies, but you might actually also get a return on your investment as well.
Okay, Pete, we have about 25 or 30 more questions, but we're coming up on an hour. And I think there's a lot of enthusiasm. Thanks for a really stimulating overview of everything that's going on. We look forward to the future. And I want to thank everyone for tuning in to the Front Row, and we will see you next time.